For many years now, researchers like myself have been studying all the possible remedies that could relieve human suffering and save lives from the deadly disease that is malaria. Today, malaria continues to be a major threat to human health, with the parasite continuing to develop resistance against all the available drugs. We are in a race against time to find the best cure. And it is in this battle against the parasite that I embarked on a journey to explore what has throughout most of human history been the most effective cure for malaria, the cinchona bark. I am Nina Rønsted, a professor and passionate nature explorer, and with me on this journey is a team of experts in botany, chemistry, microbiology and bioinformatics. Before we set out to explore the forest, our work started back home in the herbaria, where we looked through old collections, databases and literature to find the best places for collecting plant samples. Our mission was to discover places where the early plant hunters had not been and where there might still be hidden diversity left to be explored. And while our expedition took us to Colombia, Bolivia, Peru and Brazil, my first trip was to London to meet the author of the book The Fever Trail, Mark. In the book, he describes the hunt for the malaria cure and the cinchona tree. The book is a masterpiece and became my inspiration to go in search of the best cinchona bark.
Hi, Nina, very pleased to meet you. Come in. Indian peoples who live in the rainforest know the name of every tree, and they knew that it would work against this disease called malaria, and it would interrupt these fevers and you would be cured. And one Indian in particular called Manuel Inca Mamani was something of a sort of a naturalist, very good at spotting, you know, the different species, and he knew exactly where the most valuable stands of Calisaya grew. So his knowledge, as it turned out, proved to be the key, the solution to obtaining the seeds from the species of Calisaya, which, as it turned out, yielded more quinine than any other species. It's thanks to, you know, this Bolivian Indian that quinine was there to treat people and save lives.
These plants have been in our nature for over 100 million years, and over time, they have diversified and evolved new species. Through evolution, plants can adapt to environmental and ecological changes and protect themselves against herbivores, insects and diseases. In the process, they have evolved highly sophisticated defense mechanisms that we are now using for medicine. So there is a lot we humans can learn from nature.
Oh, so we're going to see some Cinchona collection.
Yes, before leaving London, I had to make one more stop at the Royal Botanic Gardens Kew to meet Mark Nesbitt. Mark curates one of the world's largest biocultural collections, and he has samples of the original seeds collected by Mamani. And if I'm lucky, I will be able to get some for my research.
And here we have Calisaya. It's a very quinine-rich swarm, and it was the basis of an enormously successful plantation export industry in Java.
So this is actually a part of Mamani's seeds?
Absolutely. So of course actually collected by his native informant.
That's very exciting.
Yeah. Would you like to take them?
Oh yeah, I'd be honored. Thanks.
Shut up.
The samples collected by Mamani will be used to do DNA analysis to see how they're related to the samples we will be collecting on our expedition. At the same time, we will also learn more about the origin of the historic samples, and in this way make our contribution to the famous history of the discovery of quinine.
And then this is the materials supply agreement. Great. Thank you very much, and it'll be really interesting to see what you get out of this.
I look forward to come back and tell you about the results soon. The real adventure starts now. Our journey begins in the highlands of northern Peru, in an area called Abria Patricia. This area is known to have several cinchona species, including the rare Cinchona parabolica, which we are hoping to find. We have brought an international team of researchers, both from Denmark and Sweden, as well as our local collaborators here in Peru. So we have botanists who know the trees and the forest, and we also have chemists along, who normally work on the bark and the alkaloids in the laboratory.
It's a new species.
Yes,
this one is
but on this trip, they're here to see the trees in their natural environment and make new observations, which will help us better interpret the results in the laboratory. Unfortunately, we didn't find the Cinchona parabolica today. We did, however, make a new discovery. Today, one of our great collections was a dokumentarfilm species that was found by one of our chemists, Sten Honoré Hansen from the University of Copenhagen. After collecting samples of the new dokumentarfilm, which is one of the close relatives of Cinchona, we move on and continue our search for Cinchona and to try to understand how the environment has influenced the chemical diversity of Cinchona trees. If we are lucky, our results will help us provide an answer to a question that scientists have been struggling with for hundreds of years. Why is it that some trees are much better at producing high levels of quinine? To answer this question, Carla Maldonado, a botanist from Bolivia, has researched and mapped out most of the Andes and has talked to locals to find the best locations for our search.
Get you so we can put a name on you and see whether you've been eating any quinine.
Alexandra Antonelli is a professor in botany from the University of Gothenburg in Sweden. Originally from Brazil, he has a huge knowledge and interest in all nature. Alex and I have been working together for many years now on our quest for Cinchona.
There are many insects that are only active at night, which you never see at day because they're hiding somewhere. And by coming back at night, you'll be able to get a full understanding of what may be attacking these Cinchona trees at night.
Also part of the team is microbiologist Chris Barnes. Besides studying insects on the Cinchona trees, Chris is also looking into the micro-universe in the soil and in the bark and leaves of the trees to understand what role the microorganisms may have on the production of quinine.
We think that the reason why Cinchona produces different amounts of quinine is purely because the environment is affecting it in much the same way that we're all products of our environment. If a tree is angered in a certain way or has certain conditions, then it will produce more quinine than other things. And so we're trying to see if there's any relationship between the soil conditions and the quinine production within the tree. So the approach we are taking to try to find new leads from plants is to take advantage of the technological development that has occurred since the early plant hunters went out to discover new drugs. We have combined chemical analysis with analysis of the soil and the organisms there. We're looking at insects. We're also trying to put all this in perspective with the genetic profiles of the plants. This is only possible through this interdisciplinary approach, where we have botanists, chemists, microbiologists and bioinformatics working together in the field and in the lab in trying to pose the right questions, find the right material, do the right analysis, and most importantly also in trying to interpret the results. As our quest for Cinchona continues, we have now traveled over 2,400 meters down from the highlands, where we found a new species growing at this altitude. This is significant, as it is the highest record of Cinchona species ever collected. It is late as we arrive at a village near the Amazonas. Traveling and collecting plants in these areas involves having a permit from the locals. So before we move on, we get the permission to take the samples we need. We've been using the river to travel down to look at the forest because it's a very easy way to get around here. The local people are using the river for transport of goods and we are using it for the purpose of science.
And what we are in particular hoping to find down here in the rainforest is the lower limit of where Cinchona species can grow. We can be spotting trees from the boat, and then we can see when the forest looks interesting, we can make small hikes into it. And also what is very important for us to look for here are some of the things closely related to the Cinchona species, which may have very similar alkaloids but slightly different and maybe even more interesting, because they are our window back in time. It is not possible to know what was happening exactly millions of years ago in the evolution, but by having these close relatives in our sampling, we can use these modern DNA techniques to actually have an idea about both the timing and what direction of events happened in the evolution. As we got caught up in the sampling process, we lost track of time, and before we knew it, it was getting dark. Traveling by boat after dark in these parts can be very dangerous due to heavy currents and sandbanks that are not visible in the dark. But we made it home safe. Our quest is not just about collecting Cinchona plants, but we are also trying to retrace the history behind the original Cinchona collections. This is important for us when trying to interpret our own data. We arrive at a village called Cascaria, which is also the local name for the Cinchona bark, and we wanted to find out why and how the village got its name. We met a lot of people who were very happy to tell us about their village and the history of it. When the villagers first came there about 50 years ago, the whole area in these hills were covered more or less with Cinchona trees, and they found it natural to name the village after these. There are not so many trees in the area now because many of them have been harvested and used, but the villagers have been very successful in making small plantations and are actually now growing the Cinchona species. And this is actually one of the things we are hoping for with this project. If we manage to find the best Cinchona species, we can potentially suggest people to actually grow these more productive varieties or species, and this will also help sustain the wild populations that are under threat in many areas nowadays.
By the age of empire, in the middle of the 19th century, it becomes very, very important for the different European powers to obtain a secure supply of this miraculous compound. So a series of botanists, collectors, explorers are sent to the Andes, to Ecuador, Colombia, Peru, Bolivia, to try and find the species of Cinchona which have the highest percentage of these valuable alkaloids. There were tremendous challenges that had to be overcome crossing the Amazon, or if you arrived on the Pacific coast, finding a way up into the intermontane forest below these majestic volcanoes that line the Andes. And once you arrive there, trying to locate the species of tree among all these different trees, which look very familiar, and trying to find the specific tree that might have large amounts of quinine in the bark. So these collectors faced a series of physical and logistical and also scientific challenges to try and obtain this plant.
Despite modern means, getting around in these areas can still be complicated. The Andes stretch across 7,000 kilometers, making it the longest mountain range in the world, with an average height of 4 kilometers. So it takes a fair amount of planning to get around here. This morning we made a strenuous hike up the hillside above Chulimani. And And we came especially to this hillside because this is the place where only a few years ago we find Cinchona trees with the highest level of quinine. However, it's a little bit of a sad morning because we haven't found any Cinchona trees at all. What we have found is a lot of fields, a few coffee fields, but primarily coca fields. And this is a picture that's emerging everywhere we go. Coca fields slowly taking over the natural forest as a livelihood for the people who live here. We knew that they had been over-harvesting at some point, and this is very sad to see, because what we know is that of the maybe 400,000 flowering plant species out there, there's only maybe 5 to 10% that have been investigated for their bioactivity or content of natural products. So this means that there's still a huge diversity out there that hasn't really been explored for potential new drugs or leads. And if we fell the forest, we don't only lose the Cinchona trees, we also lose many of all these other plants that might not even yet be discovered to science, or may be known to science, but may not have been investigated. Our quest for Cinchona trees has taken us far and wide. At one point, we are facing the prospect of driving along what is called the Road of Death, one of the most dangerous roads in the world. The biogeography of the Andes is very special, with many high mountains and low valleys, presenting a barrier for species to migrate over long distances. This results in a large amount of diversity in one area, which is why so many Cinchona species are found just here.
I can't see any.
Yeah, it's going to be a difficult one, unless you break off the whole branch further in.
Es la de atrás, es la de arriba, más arriba.
Next one has flowers.
Yes.
Right, yeah. Eso ya no importa.
Yes. Look at that. Wow. Yeah. So we have to be careful with the fruits. They are really falling off very easily.
Yeah.
Just make sure we don't get that one lost.
It seems that we have found Cinchona parabolica, one of the species we're really looking forward to find. We travel all the way across the Atlantic to South America to find this species. It has been found very few times and it's very poorly known. We have no idea about the variation within the species. Yeah, so at least 3 open flowers and some flower buds. That's excellent. We make a very nice collection.
It's really a very important specimen for our collections, and not only for our collections but for science in general. It has both flowers and fruits and all the details that are necessary to identify it without any doubt.
They also have a GPS coordinate, so we know exactly where we found it.
So we need a relatively young piece of leaf. The green ones are usually the one that has the highest content of DNA. We put it in this bag, which is full of silica. The quicker we dry the leaf, the better the DNA is kept, and so we get better analysis. So now that we have this perfect exemplar, we need to have the other data collected. So we need samples from the bark, and we also need to have soil samples. Perfect. Now we're ready to climb down again, and all that we're missing now is the soil samples. So here's some soil. It's full of roots, but we can take the roots out later when we have dried it. Now we have a complete sampling of this rare Cinchona parabolica. Now, guard it with your life. The whole expedition has been a success, and we are very excited about our discoveries. And all that is left now is to pack our samples carefully and celebrate our big achievement.
So, cheers to you. Yeah.
See what has just arrived.
We managed in these last 3 years to collect about 30 different species, more than 130 different samples, so that represents about a quarter of the diversity of the tribe. And it definitely gives us a very good starting point for understanding the diversity, what causes it and what might be left out there to discover. We took all these specimens back to the laboratory and they were investigated for their chemical contents. In this we discovered many things. Of course, very interestingly, we found that the species containing the absolutely highest content of quinine was indeed Cinchona callicea, the species originally collected by Mamani in Bolivia. We found that different populations from different places, from different mountainsides, contained different amounts of the alkaloid. We sampled in several places, more than 20 different locations, and we explored the chemistry and the genetic diversity of these different populations. One of the things we found in the field was new species of Cinchona, completely new to science. One of them is already described, Cinchona andersonii. We found that in Bolivia. There's also other species found in Peru that we are now trying to describe. So there are things out there that have never been discovered yet.
This is the total iron chromatogram.
We've also found a lot of peaks of minor alkaloids that we don't know yet what are, possibly new compounds. And combining this knowledge, we are able to make predictions about in which lineages, in which places that we are most likely to look for new compounds or for higher contents of the compounds we already know. So finding new drugs from plants is not only a historical science, it's also a science very alive and very relevant today. We know that maybe 25% of the drugs we have today originate from plants in nature. We also know that actually almost half of the medicines we have today have been inspired by nature. And it's really a great opportunity for us to be able to take advantage of this chemical and biological diversity that exists out there. We have lots of medicinal industry around the world trying to help solve these medical needs of today, trying to find new drugs for us. Many of them have been around for 100 years or more, but these plants have been around for maybe 100 million years. So that's a very impressive history for a drug development department, you can say.